So let's review. 11.2, what two body systems? You should have learned by now.
Bones,
Skeletal and
muscle.
muscle. So bones and muscles. Skeletal system, muscular system. Review. Your muscles can act as levers and for protection for other marine animals. So as a lever has three parts. So if we look at how our bones act as a lever, our bones act as the what part of the lever? The fulcrum and the load. The fulcrum and the load, those are your bones. The effort is your muscle. So together with your muscles, your bones can act as your levers. And what else did we learn? We learned the three types of joints: fibrous, cartilaginous, synovial. But we don't have to know these two, right? The only joint we need to know is your synovial joint. And these are the types of your synovial joint. But you need to focus on what joint?
Ball and socket.
Ball and socket. Our example is the elbow joint. So know the parts and the annotation of each part, description of each part. So you have to know the muscle groups, the bones, humerus, radiolna, muscles, biceps, triceps, and then the joint itself, covered by your joint capsule. Between your bones, you have your synovial fluid. At the end of your bones, you have cartilage. So synovial fluid and cartilage together prevents friction. And then the purpose of your joint capsule is to hold that joint in place. However, physical injury, this can get dislocated. This can get ruptured. Then your bones can get dislocated. All right, in your joints, because of your joints, you're able to do some movements. So types of movements are
Abduction.
abduction, adduction,
Flexion.
flexion, extension, and then inward rotation,
Outward.
outward rotation. Okay, thanks to your ball and socket joints. And then we talked about the muscles now. First of all, we learned what antagonistic means. What if one contracts, the other relaxes. Biceps contracts, triceps relax, vice versa. And we also looked at antagonistic muscles of your insects. So for this grasshopper, you have two groups. Your flexor, to flex the leg, to move it closer in a Z-shaped position, and your extensor muscle. When this contracts, the legs move farther, and then that gives them their jump. So flexor and extensor muscles in your insect leg. And then I hope I've emphasized the muscle fiber. Let's review the structure again. Before that, I did mention that there's three types of muscles: cardiac muscles, muscles of the heart; smooth muscles, muscles of your intestines, small and long, small and long intestine, small and large intestine. Why did I say long? Anyway,
It is long.
yeah, but small is longer than large.
Yeah.
Anyway, the third type of muscle is your skeletal muscle. So these are the muscles attached to your bones. And I felt like this was the best diagram. So you have your the actual muscle made up of muscle fibers. And this muscle fiber right here is made up of even smaller cylinders called myofilaments, right? So this is the muscle cell. The muscle cell is called a muscle fiber. The muscle fiber is made up of myofibrils. The myofibrils are made up of myofilaments. So these three levels, please note. Myofilaments together make up myofibril. All of the myofibrils together make up muscle fiber. In your notebook, muscle fiber, arrow, myofibril, arrow, myofilaments. Okay. We needed to know that because if you look at the structure of your muscle cell, so this is your muscle cell, under the microscope, it seems to have striations. Maybe another word for striations. You just think stripes. I like stripes better. It's striped. Dark, light, dark, light. And if we look at that, we're looking at one myofibril right now. This one, several myofibrils. So looking at one myofibril, it has a light, dark, striation. What you needed to know here are the parts of your myofibril, anatomy of your myofibril. So easy to remember, light band, dark band. In the middle of light band, Z line. That's all. And then Z line to Z line, that's your sarcomere. What is a sarcomere again?
One Z line. One Z.
The distance between Z lines, but what is it? It's the it's contracting unit. It's the unit that contracts. So along the length of your muscle, along the length of your myofibrils, you have sarcomeres. So all of those sarcomeres will contract as a unit. Together, you've shortened the whole muscle. I hope I can have like a model. You know, like you can collapse so you can see all of the sarcomeres becoming smaller. I hope they have like a model we can buy. Okay, but I hope you understand. Now, under the microscope, it looks something like this. So, clear striations. The Z line, very obvious, right? Now, what causes those striations? The reason is your myofibril is made up of myofilaments. Two types of myofilaments, myosin and actin. So myosin is the thicker protein molecule. That's why it looks darker. Your actin is a thinner protein molecule. That's why it looks lighter. So the area where they overlap here is the dark band. So the dark band is made up of myosin and myosin overlapping with actin. The light band is only made up of actin filaments. Okay. So knowing that anatomy of your sarcomere, we have to be able to explain muscle contraction. Now you cannot explain muscle contraction if you don't know the structure. That's why we have to learn the structure first. So this is your structure of your myosin. Light band or dark band? This picture. Dark band. There's an overlap, right? Area where they overlap, that's still part of your dark band. So this area right here, if I zoom in, it's this second picture. You see that there are heads sticking out of your myosin, and then you see your thinner thread-like actin. And let's try to review how contraction happens. So in a nutshell, just look at the head. The head at first is touching the actin. You use ATP, the head separates. Hydrolysis of ATP moves the head to a farther location. Hydrolysis of the ATP moves the head farther, and then it touches a farther location in your actin. And then removal or release of your ADP results into power stroke. So you need your ATP twice. First, you need the ATP to remove the myosin from the actin, right? Second, you need ATP. Hydrolysis of ATP will result into movement of the head to a farther location, and then power stroke when the ADP is released. So you have to look at this part, and this part, and this part. On the when you hydrolyze ATP, it becomes ADP and P, diphosphate and phosphate. You lose the phosphate. That's not a power stroke yet. Hydrolysis of it just moves the head to a farther location in your actin. then it touches a new location. And then the removal of ADP, that's the power stroke. Here, removal of just phosphate. Only phosphate is removed, myosin head moves. And then if you remove ADP, boom, power stroke. If we want to be very technical, listen, everybody. Use ATP, remove the head. P is removed from ATP, just P. Move the head, touch a new location. ADP is removed, head moves inward, power stroke. Okay? Again, on the test, the question will go, how do you use ATP in muscle contraction? So ATP to remove the myosin head from the actin, and hydrolysis of ATP results in movement of myosin head. Removal of ADP results in power stroke. That's why your muscle fiber contains a lot of mitochondria, because it needs to make a lot of ATP.
First,
Okay.
ATP detaches the head.
First, ATP detaches the head. Second, it's hydrolyzed, it moves, it touches. ADP is removed, boom, power stroke. So possibly 3 bullet points there. All right, let me show you some animations first. Why do you want to know? Diagrams here. So familiarize yourself with the bands first. Oh, by the way, those are the different parts that the previous batch needed to memorize. Remember, for you, just Z line. So lucky.
Wait, miss, the heads aren't all over the myosin.
No, the ends of the myosin will have heads.
So the middle part doesn't
No, only the part where they overlap. That's
If
what we
they-
predict. That's just our model. But again, we really don't know.
Yeah, but if they are attached side by side, one contracts, how can the other contract?
Together. Like
So-
if we're 2 sarcomeres, 1, 2, 3, then collectively the whole muscle length. Again, this is just a proposed theory. That's why we call it sliding filament theory. Maybe it works like this. During my time, it was not sliding filament. It was walking filament theory. It's like walking. The myosin head is moving through the actin. Sliding is just one power stroke. So, anyway, we can expect to have more possible theories in the far future. But for now, this stands to be the most accurate. All right. So, how many sarcomeres do you see there? 2, right? This. So what happens is, no explanation, just visualize it, please. So the myosin, those are the different parts, and contract. Well, hold on, I have other animations. Just to show you, like, collectively what we did, Sally. The whole length shortens. Now, if the muscle is contracting, do you still see the... this is previous batch. If the muscle is contracting, the H zone disappears. But you don't have to know the H zone. Okay, let's look at another animation. So right now, you know the shortening. But the question is, what happens before that? Why did the muscle shorten? So here's the explanation. Your muscles are innervated with nerves. Innervated means there are nerves connected to your muscles. Your nerves are coming from your brain. So if your brain wants to move your muscle, it sends a signal through the backbone, through the nerves and into your muscle. to trigger your muscle to contract. Now, that nerve obviously needs to send, remember in 6.5 we learned the nerve impulse. There should be a nerve impulse first from the nerve. So imagine that there's a nerve here. There's a nerve. And then what does the nerve release? Neurotransmitters. So the neurotransmitters will bind to your muscle cell, causing another action potential in the muscle. So this muscle will have an action potential. The action potential will reach those orange organelles. We call them the sarcoplasmic reticulum. So the action potential will spread through your muscles. Oops, what just happened? Please look, Cheska. The action potential caused the sarcoplasmic reticulum to release calcium. Again, action potential spreads through your muscle, causing your sarcoplasmic reticulum to release calcium. Why do you need calcium? Let's look. What does calcium do? The calcium will go to your actin filament and, please wait, zooming in, what happens is your calcium binds to your, where's my calcium? Troponin. You remember last time I told you the actin is made up of 3 protein molecules: troponin, tropomyosin, and F-actin or actin. This is your troponin, this is your tropomyosin, and then this is your actin. Which is purple actin, this thread tropomyosin, this red subunit troponin. Now calcium will bind to troponin. Binding of calcium to troponin results into change in the position of the tropomyosin. I don't know if it's clear, but do you see what just happened? When I moved the tropomyosin, what did you see? These ones. It was hidden before. Look. Hidden. Those are the binding sites. Those are where your heads can bind. Now calcium, again, will bind to troponin. Troponin will change position, and as it changes position, it moves the tropomyosin, exposing the binding sites.
So Miss, can the myosin can't bind to the other part?
The myosin head cannot bind to these ones unless it's exposed. So backtrack, we need calcium. No calcium, nothing to pull this away to expose the binding sites. Now it's exposed. We've learned this already. Now it's exposed, your myosin heads can now bind and cause your power stroke. Easy to understand, right? Easy to forget too. So maybe in your notebook, put a diagram, action potential, arrow, calcium released by sarcoplasmic reticulum, arrow, calcium binds to troponin.
Action potential.
Summarize it the way you'll remember it. Just arrows. Action potential, sarcoplasmic reticulum release, calcium. Calcium. Quick, just do legends or keywords. Calcium binds to troponin. Troponin moves the tropomyosin. And then next, myosin can bind to your actin. Finish. Put a smiley. Tell yourself, I will memorize this stuff. Clarice, talk to your cousin. Clarice, talk to your cousin. I don't care about word per word. I want you to understand the general idea.
Miss, it just moves the tropomyosin. Doesn't it move the whole actin?
No, it moves the tropomyosin. The tropomyosin is covering those binding sites. So it has to pull it away to expose those binding
Miss,
sites.
but the action potential part, I don't really get action potential
There's
to
a-
sarcoplasty.
Oh, the action potential is a charge, right? It means the muscle is now charged, positive.
Neuron.
So that charge tells your sarcoplasmic reticulum to release calcium. If if the charge reaches the sarcoplasmic reticulum, it stimulates it to produce calcium. All right, this is now the animation for the ATP that we just learned earlier. So remember, it's used twice. So first, hold on. So at first, it's binding. Let me see. No. This doesn't have the first stage. Okay, what's the first stage?
ATP.
Removes. This one doesn't have. So ATP detaches the myosin head. So this is the second part now. You have that ATP touching your in your myosin head. Then P is removed. If you lose the P, it touches a binding site, right? And then if you lose the ADP, you have the power stroke right there. Pew, power stroke, power stroke, power stroke. So binding of the ATP detaches the head. Release of the ADP causes the power stroke. There. Since it's a cycle. Okay, watch with me. So ADP is your loss of ADP is power stroke. So it's still sticking there. Your muscles don't remain shortened. I mean, I don't want my muscles to remain short. I want it to put it in the original position. What do we need? ATP. ATP will move the head. There.
Miss, what is cramping?
Sorry?
What happens when
Muscle cramps is when you don't have enough oxygen circulating in the muscles. So the muscles, if there's not enough oxygen, you don't have enough ATP. Remember, you have anaerobic.
It's just sticking together.
No, it's still contracting, but it can't keep the muscle cramps, the pain is not because of the movement. It's because you're producing lactic acid in anaerobic cellular respiration. There's lactic acid in the muscles now. Then you feel the pain. Obviously, if there's pain, you can't keep using your muscle. But it doesn't mean your muscle is contracting, like it's not going to its relaxed position. It's there, but you can't move it because lactic acid is triggering the pain response. Okay, so I hope that's clear enough. ATP detaches. ATP detaches, please look. Hydrolysis moves the head. Removal of P, this touches another position. Removal of ADP, power stroke. And then ATP sticks to it again. If calcium, if there's no calcium, nothing will happen. So not only do we need calcium in your bones to strengthen your bones, you also need calcium for movement. No, we get it from our diet. It's a mineral. So our food, when we eat food, there's calcium. We eat fish, there's calcium. We drink milk, there's calcium. So it's the food that we eat. So good question. We don't make it. We get it from our diet.
We get it
from
water.
Yes.
Miss, but
No, but what?
okay.
Okay.
Removal of P, before the removal of P, the head is
Before this,
it moved already, the head.
simultaneous. This is simultaneous. Hydrolysis. Remove boom. As it's removed, as it's removed, it's touching.
But like, can you go a little? Head was here, right?
No. Yeah. Head was here. Let's start with that.
Yeah,
Head
and
was
then-
here. This is simultaneous. It's flowing.
Yeah.
Don't go technical part per part. Bind, remove. It's being hydrolyzed. It's hydrolyzed now. It's moving it moved the head.
Yeah.
Then loss of the P touches
But just the
the actin, and then loss of the ADP moves. Everyone, that was a good review from Sally. Please look one more time. We've discussed this a million times. It's okay. So, please look. ATP moves the head. ATP is hydrolyzed into ADP and P. It moves your head. P is removed. It touches the... So long. P is removed. It touches the binding site. ADP is removed. It shortens. Power strip. All right. So NOS. So how does science work? How do we know that we need calcium for your muscles to contract? We have to know 3 species are examples. Here, can I borrow some? So the first species is your jellyfish. 484, please go to 484. So your jellyfish has a special protein that is sensitive to calcium. So when calcium is present, this protein is bioluminescent, it lights up. If calcium is present, the protein lights up. So they removed that protein from the jellyfish and they injected it in an acorn barnacle. You know what a barnacle is?
Acorn is the one squirrel eats.
Yeah, well, it's maybe the shape, that's why we call it acorn barnacle. Looks like an acorn. Like rocky shore. Along your rocky shores, you see the sticking on the... Can you turn off the lights here, Chloe, so you can see?
It looks like fungi.
Yeah.
Both.
Huh?
Both lights.
Disgusting.
So in rocky shores, they grow in the rocks.
Oh.
They grow in the rocks. And if I look at anatomy of barnacles. Bam, I miss you, I love you, but I'm lecturing. I want to see you later. Bam, yeah, it's Bam. Bye, Bam. See you later.
Umbay.
I'm sorry, Umbay. Why was I saying Bam?
I don't know, you're not even in your class.
I know. Maybe because... Sorry, Umbay. I'm so happy.
I was rolling with it.
Thank you guys for not telling. Umbay, hello, Umbay. Maybe because I'm friends with the twins and I always see them in my Insta stories, and I've been thinking about if the twin and Bam is still together because I don't see pictures of
They
them.
are.
Okay, good. That's why I'm thinking of Bam. Umbay, I miss you. Like really, you, Umbay, not Bam. Later. You know what I like about Umbay? He remembers things. The first thing that I told him when he was a new student, I actually forgot about it. And then he's like, Miss, you remember you said this. I don't remember. And then when he told me, I'm like, yeah, I kind of told you about that. Anyway, so he has very good memory. Okay, so this is the anatomy of your barnacle. Oh, by the way, this lecture is being recorded, so whoever's going to listen, excuse the commercial. So this part of your barnacle has muscles. This opens and closes to allow movement of water in, so that your barnacle can get nutrition or food from the water. So this muscle moves. What they did was they injected the bioluminescent protein from the jellyfish to your barnacle. Now what they discovered was when it moves, the protein lights up. So what's the conclusion? When the muscle is moving, it was lighting up. Remember, it can only light up if
Calcium is
there's
present.
calcium. So that's how they kind of made the hypothesis that we need calcium for muscles to contract. Because when this contracts, it lights up. It can only light up if there's calcium present. That's it. That's the NOS. So it says fluorescents have been used to study muscle contraction. Again, these are just models, but we also did some experiments. And we know that calcium is needed for muscles to contract. Okay? All right, brain break. And we'll proceed. Wait, let me pause my recording.
I was like, is that fun?
Yeah, first she even shouted and she was like, ah!